So today's webinar will be recorded, and you can go to the YouTube channel and you'll see the recording there later on today probably. And there's also a variety of other webinars from drug design and docking and molecular modeling there as well, and other tutorials that you can view. If you just search MolSoft or MolSoft Help, you'll find those. If you're not familiar with MolSoft, just a very brief introduction. We're a San Diego-based company, and we were founded in 1994 by Ruben Abigan, who is also a professor at UCSD. Max Totroff is our principal scientist, and Eugene Rausch is our principal developer. You may hear me referring to ICM, which is our main software. ICM stands for Internal Coordinate Mechanics. And there's some papers describing this method approach that I can send you if you're interested in reading more. But basically, for our coordinate system, we use internal coordinates rather than Cartesian coordinates. We also have a biased probability Monte Carlo method for docking and modeling. We have a variety of cheminformatics tools and ligand-based screening tools as well, including atomic property fields. And over the years, we've collaborated and partnered with big pharma companies and academic institutes for development of software and the use of our software for drug design. More recently, we've collaborated with Enamine and the NIH on development of giga-searching of large billion chemical size databases. And this year we released a new scoring method called a neural network score, which we'll look at today. And over the years, we've always competed in docking challenges and modeling challenges. And the most recent one, the D3R competition, we've outperformed other methods for ligand docking and screening and prediction of binding affinity. I can send papers on these if you're interested, just get in contact with me. So the ICM family of products, we provide with this webinar, if you're not already a user of ICM, we provide a 30-day key, so you can basically follow along with what we do today. If you get the key, you'll get all these features basically up to a point. So it's called the main software modeling package is called ICM Pro, which you can place all these products inside this one package. So you'll receive ICM Pro with ICM Bio, which is a full bioinformatics suite, which we won't cover today, but there's webinars on that, where you can build sequence alignments and link those to structure. There's a full suite of cheminformatics tools in ICM Chemist, so you can read in SD files, calculate properties, build chemical spreadsheets, plots, SAR analysis, things like identify activity glyphs, for example, also build QSAR and 2D QSAR models in that. Most of our customers that have ICM, they're comp chemists and comp biologists have ICM, and then they can share what they produce from their experiments with their colleagues, who can just download the free visualization tools. So anything you do here, you can save, and you can view in iMolView, which is a free viewer and structure analysis tool on Android or Apple mobile devices, iPad, for example. ICM Browser is a tool for Mac, Windows, and Linux. And ICM JavaScript, basically you can save most things in ICM and display them in a fully interactive web page using ICM JavaScript. Some new tools that are available, we had webinars on each of these last year, and I can send links to those if you're interested in looking at them more closely. But MolScreen is a collection, a large panel of AI models for a variety of drug targets and ADME-tox targets. GigaSearch, we developed with collaboration with Enamine for being able to substructure search giga-sized libraries like the REAL database or the NIH's SAVI database. And you can do fingerprint or substructure searches are there. And RIDE is a new tool where you can screen about 500,000 chemicals per second per GPU. And it's sort of it's a fast ligand-based searching method. So your chemists could have this JavaScript window here and just sketch a lead compound and then do a search of a larger database in minutes or even quicker, and then generate ideas that way through RIDE. That's just an overview of the topics. So today's webinar topic, we're focusing primarily on the 3D ligand editor. I'm going to show you how to set up your ligand and receptor in the editor. Generally speaking, the editor is used if you have a co-crystal structure or a model with your ligand already bound. That's generally the scenario that you would use it for. You would use it for optimizing that ligand, optimizing the potency, improving specificity, things like that. You can actually use it if you have an empty pocket. You can place the ligands inside as well. That's not. You can also use the ligand editor for that. But in this example, we'll be working from a co-crystal structure. So we're going to look at how to display the protein pocket, the binding pocket, look at the properties of that pocket, look at regions where you can build from your molecule, add substituents where substituents would fit, things like that. Look at ligand strain, how to minimize your ligand, your design that you made, and also how to edit the ligand in 2D and 3D. And then in the coming weeks, we'll look at sort of more advanced or more, other methods that are inside the ligand editor, for example, covalent docking, induced fit docking with multiple receptor conformations to represent flexibility, for example, ligand-based modeling inside the editor. And then in the third week, we're looking at things like group replacement, core replacement, and isosteer searching. So the 3D ligand editor, which we're going to cover today, as I mentioned, is developed by MolSoft in collaboration with medicinal chemists at Novartis. So ideally, it's quite user-friendly to use. You don't really need to have too much training. It's just a way of exploring ideas. So you can read in your ligand co-crystal structure here, and then you can just play around with different substituents, screen substituents, and everything is you can undo and redo and save to a chemical table and things like that. So it's a way of exploring your chemical intuition and also designing new lead compounds. If you want to read more about the collaboration between MolSoft and Novartis, there's a paper here which describes the development of this along with other tools there. So today's example that we're going to use is a bromodomain called ATAD2. So bromodomains are acetyl-lysine specific protein interaction targets, and they play a key role in regulating gene transcription. The one we're focusing on today is ATAD2, which contains a bromodomain, and it's a target for oncology because increased expression correlates with poor cancer outcomes. So there are other bromodomains, for example, BRD4, which are in the BET family of bromodomains, and looking at those structures and other data, it sort of indicates that there's regions around this pocket away from where the acetyl-lysine binds, that you can sort of improve the potency of the ligands and also specificity in the family of bromodomains as well. So one of those regions is called for example, in BRD4, it's called the WPF shelf, which is around this region here. And for the example that's for example BRD4, but for our one that we're looking at, ATAD2, the shelf has different residues, RBF, in this shelf region. So we're going to sort of utilize that area as we go through the webinars to try and improve potency and specificity. And there's an excellent paper by scientists at GSK where they solve these bromodomain inhibitors. There's additional papers beyond this as well. But in this example, we're going to be using PDB5A5Q. And there's also others in this series, in this paper. So you can play around with modifications in the ligand editor and then compare them to the crystal structures. Okay. So our first step is to obviously read in the PDB file and then set up the ligand and receptor. So I'm going to go to ICM now. If you have ICM on your machine, you should see an icon like this, which you can just click on and it will load up ICM. I have already loaded the 5A5Q. So to do that, you would go to I've got this fancy new magnifying glass. You go to the search button here and just type 5A5Q in the PDB search, and then you can just press this binoculars button and it will load your structure. So then the structure will be loaded, as you can see here. The A chain is the protein chain, so you just toggle that display on and off. There are additional het atoms and other molecules that are used in the crystallography, which we're not particularly interested in, but the ligand is this is the A6XC ligand, which is shown in orange here. So the key interactions we can see is that they have this basic piperidine substituent here, which makes an interaction with this aspartic acid. With the, just move the thing now. So the aspartic acid 1071 here, shown here. And this is the region where the acetyl lysine would normally bind. And so this sort of mimics that position, this inhibitor. So this is our initial inhibitor that we're going to look at. There are lots of webinars online about how to manipulate structures in ICM, but generally rotation, the tools that I'm using for rotation are here by default. Certain parts of the screen are devoted to different actions. So for zoom, you can zoom in or use your middle mouse button. Okay, so we first need to set up the ligand and receptor in the ligand editor. So all the ligand editor functions are in this ligand edit tab that you'll see at the top of your screen. If you single click on that, you will get these two buttons here. The first one is for setting up the ligand, and the second one is for setting up the receptor. So we'll start by setting up the ligand. We click on this button here, and ICM asks you some questions. Which one is the ligand? So we actually have some other molecules here which aren't of interest. But as I showed earlier, the ligand of interest has the code A6XC. So you can see here A6XC and A6XC here as well. And so this is the ligand we're going to set up. We can choose to auto assign formal charges here using MolSoft's pKa model. You can also, in the ligand editor, optimize peptides as well. So you can, if this was a peptide, you would recognize that it's a peptide molecule, and it would enable you to edit it. So just go OK. So now you can see that in the ligand, in the ICM workspace, we have two objects now. We have our original PDB file, 5A5Q, and then we have this additional object called 5A5Q_lig. And so this is the ligand we're going to edit. So now we need to set up Untitled ICM, which is the receptor. So we click on the receptor button shown here, the second one along, this one, and it loads this this table. There's two tabs, so you can, as I mentioned, we'll cover the APF one later in the next webinar, but there's two options. You can optimize your ligand using structure-based approach, or you can use APF, which stands for atomic property fields, which enables you to optimize, design new leads based on 3D pharmacophore properties rather than necessarily the protein structure. But you can use them in tandem, but we'll talk about that next time. But we'll just be concentrating on protein. So first question is, what is the receptor? The receptor object is 5A5Q. And then we have some important questions about the waters. So generally you'd assume that the ligand may displace most waters, but obviously there are some waters that are tightly bound to the pockets, which may be maybe 3 or 4 hydrogen bonds with either a ligand or the receptor. It's kind of subjective. You need to sort of inspect the pocket and see this. If you check this, ICM will automatically keep the tight waters, or you can choose just to delete them. But you may want to inspect the pocket and see which waters you may want to keep or not. Okay. We then have some questions regarding preparing the receptor. So we just read in a raw PDB file. So there's no hydrogens on the PDB structure and the crystal structure. We can see that you can see here that it has, you can see it says 2 angstrom resolution, so it's a reasonably high resolution structure. But we still need to add hydrogens to that. So we need to, if we added them, we need to optimize the hydrogen bonding network. There may be cases for particular residues such as asparaginins and glutamines and histidines where the crystallographer cannot sort of determine which orientation that they're in. And so ICM will optimize the orientation of these residues based on the best hydrogen bonding network and try and fit them. If you have a very poor resolution structure, you may have side chains in your pocket, in your ligand binding pocket, where there are atoms missing, the occupancy is low. If that's the case, you would definitely need to be careful trying to use an in silico approach to docking there. But you can either uncheck this, and MolSoft ICM will build the full chain of the residue in the binding pocket, or if you uncheck this, ICM will just leave the area blank, leaving no additional atoms in that region. So this if you uncheck that, it will build the full chain. But in this example, we have a high resolution structure, so the occupancy of the pocket is very good. If you look back at other tutorials, you can see in the display, there's different ways of visualizing occupancy and B factors and things like that in your protein structure. Okay, so now we have to tell ICM where the pocket is located. We have inside ICM a tool called ICM Pocket Finder, which basically takes a probe and looks across the surface of the protein, and it will find cavities for you to dock to. And the cavities have properties assigned to them, and such as volume and hydrophobicity, things like that. And then they're ranked by how drug-like they are. But usually in the ligand editor, you are starting from a position of where you have a co-crystal, so you know where the pocket is anyway. So you could say, we can use this option called make box around existing ligand. If you have, if you want to, this pocket finding could be useful if you want to find sort of a cavity, maybe a sort of an adjacent cavity you may want to build into. You could use it for that, or obviously designing allosteric inhibitors as well. So we're going to choose the option make box around existing ligand. So it's going to single click on this option, make box around existing ligand. So then you can see that they have this purple box, which represents where we're going to build the maps for docking and optimization. So you can just drag to make this box a little bit bigger. And in this case, I actually need to make it a little bit bigger because that shelf that I was talking about is actually a little bit away from the pocket. So you just need to drag and encompass a bigger area. If you just go OK, once you're happy with that pocket, the dimension you drag by just clicking on the corners and just go OK. So now it'll generate the maps and prepare the ligand for modeling. So I'm just going to quickly go back to my slides. So we set up the ligand and the receptor, and we also have a JavaScript version. So let's quickly show that in ICM. So what we're doing here inside ICM can also be done on the web. So this is a web page, and you can search for 5A5Q, for example, which we're using today, this molecule. And then the setup options that we did in the ICM can also be done using this button here. You tell ICM, this is ICM JavaScript, which is the ligand and the receptor. And then you can do the editing, which we're going to cover in a little bit in ICM. So I just wanted to point out that this is a web page, a variety of different tools. To get the ligand editor, you need to you need to go to modules and then choose ligand editor. Otherwise, it just opens in the browser. Okay. So once you have set up the ligand and the receptor, you see by default we have this this surface, which we'll look at in a minute, and you can also display a different surface, which is called the receptor pocket surface, and these are colored by binding properties. You can display hydrogen bonds, which are spheres, and the thickness represents the strength of the hydrogen bond, and the coloring also sort of indicates the strength as well. We have these circles around each bond, which sort of represents the energy, called energy circles. So if the energy is poor, they'll be red. If the energy is good, it'll be green. You can also display the relaxed ligand. So it's quite easy to start designing ligands, but you're actually adding strain to the ligand, so it's not an ideal modification that you're adding. So you can easily see how different your ligand that you designed is from the relaxed version of that ligand. You can also display unsatisfied hydrogen bonds where you may want to build towards. And you can also display these interactions in 2D. And I'll show you how to do that in ICM now. Okay, so in the ligand editor, all the display tools are in, well, most of the display tools are in this drop-down button here. So if we drop down, we can toggle this ligand pocket surface on and off. Okay, so this ligand pocket surface is useful because it helps you identify regions in your structure where there is space to build. And so you can see quite clearly at the back here, there's no space at all to build. But in this region, we have a cavity to build into. The green represents hydrophobic area. Blue is hydrogen bond donor, so it's guiding you to say that a hydrogen bond donor might be a good addition at this point. And red is hydrogen bond acceptor. So this surface sort of guides you as to what the best modification would be in terms of improving the interaction with the the receptor. The opposite coloring to this surface is called the receptor pocket. I'm just going to show that one. So this is colored by the actual receptor. So this is a hydrophobic region. This is the region where that shelf is located up here, and it's colored by the receptor. So hydrogen bond acceptors are red, hydrogen bond donors are in blue, green is hydrophobic, and white is lipophilic region. Yes, sometimes with these pockets, if you go to view and then choose sketch accent, you can see the depth a bit better with the sketch, you know, the coloring, the shading of the pocket. So we can see the pocket here. So other options are you can toggle the hydrogen bonds on and off using this button here. Use my magnifying glass. It's this one here, and so just click on that, and then you can see the hydrogen bonds that are made here with these spheres. So you can toggle those on and off. You can also visualize, as I mentioned, you can visualize the relaxed ligand here. So the relaxed ligand is in yellow. So all pretty decent, matches pretty well as you'd expect. It's in the PDB. You can display the unsatisfied hydrogen bonds, and in this example there should be one to build towards. Let me just top of that. Yeah, okay. There's not one in this example, but if there is an unsatisfied hydrogen bond, it will be you'll see it displayed in the pocket. So that could be a good starting point to build out towards, possibly. You can also display the contacts. What is the key contacts your ligand is making with the receptor? So if you click and hold here, choose Analyze Contacts. This is it's going to scale the stick representation of the receptor based on the contact level. So if it's a very high contact, the stick will be thicker. If it's a weaker contact, it's going to be thinner. Just go OK. So we can see. So you can see that this asparagine here has got a thicker stick, probably making the biggest contact with the ligand. Aspartic acid, as I mentioned, making a interaction with this the piperidine, this basic piperidine here. You can also see there's a table that is produced at the bottom, which also shows the contacts, the contact area, the exposed area, how buried the interaction is, and the distance as well. So if you just click on here, it will take you, it will select. So this is valine 1008 in this molecule. So you just double click on that, you see the selection. We have these green crosses in selection. So if I center, it's going to center onto that. That valine or whatever you have selected. Okay. In the display options, there's also a what we call pretty view, which you can just change the different way of viewing the molecule. So there's also a light tab here where you can change the transparency of the ribbon representation. Okay, so those are the main visualization tools. You can also produce a 2D plot of the interactions just by clicking on this button here, this one, and this will just go okay. But these are just some parameters for cutoffs for certain things, but for hydrogen bonding, for example. So then you get a 2D plot here. The coloring is this green shading sort of represents hydrophobic regions of your pocket and side chains. Blue shading represents hydrogen bond acceptors. White dashes here shows the distance and the hydrogen bonds. These grey parabolas represent the accessible surface area. And this broken line indicates there's no space really here to build, but you've got space here to build out from. And it's a kind of like a cutoff. The size of the labels indicates the amount of strength of the contacts involved. And you can save this as an image if you wanted to. You just right click and choose Save Export Image, and or copy it to clipboard or whatever. And I mentioned earlier these energy circles. So these, if you just click on this, just zoom in. You click on this energy circle button. You'll see that these values, the green values, represent the contribution to the binding score that we have. So if green is good, so all is well with this ligand initially. We may want, because we just read it in directly from the PDB, we should really just do a small minimization of this ligand. So to do a minimization, we have some tools here. There's the one on the left is for minimization. The one on the right is for full ligand docking. So in this example, we just read in a PDB structure. So we've added hydrogens and things like that. So we need to just minimize it to the nearest energy well, and that will just click on here and it will just do a small minimization. There you see now we have a better hydrogen bonding network. We can see that the ligand strain value was high before, now it's gone down to a reasonable level of 3. We have a reasonable RTCNN score, which is our new neural network score. So the lower these scores are, the better the interaction is. And so yeah, we have this salt bridge with the aspartic acid here. Hydrogen bonds shown here. Okay, so now we've investigated where there's space to build. We're ready to start editing the ligand. Back to this. So there's different ways to edit. We can edit in 3D. So basically, when I show you in a minute, you'll see this panel. You can change, for example, atom types. You click on carbon, click here, and it will change to a carbon. You can change bond types. Just click on the bond type and then click on the bond here, and it will change. You can select one or more substituents and then make the modifications to the point in your ligand. There are some other options which we'll cover next week, like distance restraints and tethers. And you can erase, and everything can be undone and saved, so you don't have to worry about ruining the thing. So we're ruining the structure. You can also change the charge as well. So we had a question just a minute ago about protonating that pyridine. you could, if it's not already protonated, you can protonate it using this editing tools here. So, or alternatively, in the same panel, you can just click on the 2D editor and you'll get this window here. And you can just make your changes in 2D and see those, and then commit those changes to 3D. So let's see how we can start doing that. That's going to go back to ICM and find ICM here. So to get that panel of editing tools, we need to click on this button. I'll just go and zoom in again. This button with the pencil represents the editing tools. So you just click on that button and you get that panel which I showed earlier. Just for clarity, I'm just going to delete some of these things. Don't need them anymore. Okay, so we can start editing. We're going to start initially by using the options in the 3D sort of way of editing. So as I mentioned, you can change atom types by clicking on an atom here, and then you click on the atom you want to change in the ligand structure. Just click here. You can see now the nitrogen has turned to a carbon. We can evaluate the score. The score has got higher, it's got worse because we've lost an interaction with this asparagine residue. So to start with, that's not a good modification, so we can just undo it. We use the undo button here. Let me just show magnifying glass. Undo button is here. So just go undo and it will put back the nitrogen, and then we have a good, nice hydrogen bonding network here. To change bond types, we have the bonds shown here. So we have the single bond, double bond, triple bond. So if we just click on double, for example, we can, or single, we can change this bond type here just by single click, or we can turn it back to the double bond shown here. And there's also a way to, you can also delete atoms, whatever you want to do. So there's the eraser button shown here. It's eraser. Click on the eraser, then you can delete substituents and evaluate the score shown here. So this methyl doesn't make a great deal of difference, but just put it back. So these are the options to use this panel. There are also inbuilt substituents. There's some common ones that we've placed here that we can use. So we could try, as I mentioned, we're trying to build up towards this area, this hydrophobic region, and this, not showing the residues at the moment, but there's some other residues around here that we can build towards from this, the C3 and the piperidine here. So we could try an ether linkage. Just click on this here. Just click on the panel here, and then you just click on the hydrogen that you want to make that modification to. So I don't know whether Zoom shows it, but you can see that the cursor is like a cursor key here. And then when you get to the hydrogen, you have a little finger pointing at that hydrogen. So you just click on that hydrogen and it makes that change. So you can see it did a small minimization for that when it modified it. If you want to, if you're making very large modifications, you can redock, and we'll cover that next week, in 2 weeks' time. Or you can just do a small minimization again after that one if you wanted to, just to be sure. And you can evaluate the score. So you can see this modification actually improved the score it went down. It's improved the score a little bit. So if we like this modification, we can add this modification, we can save it to a chemical spreadsheet. So we click on this button here, and it will save your design to this spreadsheet. So you can always go back to the spreadsheet, just double click and load it again. So we may want to, we seem to have found something, a good position to start trying to improve our molecules. So we could try some additional substituents there. So I'm just going to go back to the original structure, just go undo. And I'm going to screen some other ether linkages that we have here. So you can click one, as many as you want. You can click the whole panel if you wanted to. I'm just going to click these 4. And then if you go back to that position and click on the atom, it will screen those substituents for you and then rank them. So we have, you can sort the table. You can see that we have the RTCNN score here. Just ignore that column. That's this is the development version. That's I think it's a placeholder for something. But we can just sort by score. So the lower the score, the better the interaction. So if you want to see this modification, just double click, and it loads it. You can go through the table of the best ones here, and display them. Or you can just toggle if you double click, it loads the ligand into the actual receptor. But if you just want to toggle the view of something, you can just use these boxes just to toggle on and off. Or if you want to load this in, you double click. So that's in 3D. I just wanted to also point out that everything you do is recorded. So if you click and hold on the undo button, just gonna click and hold, you see that there's a little green, there's a little down arrow here. If you click and hold here, then you get a full modification history. If you click on that, you can see exactly what you've done. So we changed the atom, we used modifiers. So you can always see what you've done. You can always go back, just double click, and it will load that into your structure. So you can see this is the original PDB. Then we minimized it, and you can see it. Yeah, and if you want to, if you like a modification that you've made, you can tag them as well. You can save it as a chemical spreadsheet, or you can save and tag. So you can say, give the series a tag. Let's go see the pyridine ring here. We can give it a name like that, and then you can see that when you add them to your modifier, you should get a tag on your table, so you can sort them. So just do that again. Click and hold, save and tag. Okay, yeah, so yeah, the tag is in the first column here, so you can see series 3, C3 series. You can give, you can save them. And so if you wanted to share this with your chemist, who's going to actually build the molecule, synthesize the molecule, you can make notes here, for example. And also, if you want to keep your chemist happy, you can calculate properties here, chemistry, calculate properties. And we have an option called MolSynth, and that's how synthesizable this molecule is, how common these groups are seen in other molecules. So you're not designing something that the chemist won't be able to build, ideally. So basically, 0 would probably indicate that it's going to be a challenge for your chemist to build a design that you've sent them. I think in future versions we may include this automatically in this table. Right. So that's editing in 3D, but editing in actually while we're here, you can also just manually edit in 3D as well. So you can rotate torsion angles. If we click on the torsion button here, this one, single click, you can rotate the torsion. If you rotate, you can just undo if you don't like what you've done. If you have the energy circles displayed and the torsion button, let's just show you this gives you the angles, this VAR variable button here. So if you then use the torsion rotation button, click on that, and then when you get to a point, you'll see you have the cursor. When you get to a point where you can rotate, you'll see that you see the angle moving. If you strain, then these rings will be colored red, and you'll see that the rotation. So you rotate by just dragging. Okay, so these energy circles are quite useful. So, for example, if you try to add a group, a large group here, for example, it doesn't fit. You managed to fit it in. There's a space here, it's okay. But you will see that. Let's try something particularly large. This side as well. Yeah, you'll see red arrows saying. You'll notice in the score as well. You get a high score, but it would also flag clashes. Okay, so I've made tons of modifications. I'm just going to go back to my original. Go to the modification history. Go to this table. Go back to the original minimized Ligand. Okay, so we have these inbuilt modifiers. You can add your own if you wanted to. You can click on this button here called Add New, and you can just sketch the modifier that you want, or you can read in the table of modifiers. Let's just do, for example, nice phonomide or something like that. But you need to put an attachment point. So where you want the group to be attached, you right click and choose attachment point here. So you'll see an asterisk on the atom that's the attachment point here. So then you just go close, and it asks you want to save changes, go yes or no, and then it will add it to the panel. But you may want to be, explore a bigger set of chemistry than just these 20 or so groups that we have by default. So to do that, we have this option called Advanced Group Scan, and if we just click on there, you'll see a new window come up. So this contains 60,000 substituents that you could try. You could just click on this hydrogen and let it run at your screen, or 60,000, which might take a while to run. But or you can just narrow it down to particular properties that you're looking for. I want to find small hydrogen bond donors groups. If you click on that, that will filter this down to, you can see here, this was just screen 2,374 substituents, and you can see them here. If you have your own table, you can just choose local table and read in the table, and it will screen those, but it would need a attachment point defined. You can filter this table by different properties as well. Using this drag, you can see that this filter icon here means that this column is filtered according to your filters here. You can filter by chemistry as well. You can sketch the substituent that you want to find in here. Once you're happy with your database that you want to screen at that point, you can choose whether to ICM to add charges based on the PKA model that we have in ICM. And to speed things up, you can choose number of parallel jobs based on the number of cores that you have on your machine. Okay. And then you just go OK, and then you get a hit list similar to this, ranked by score. Okay. So you can also edit in 2D as well as 3D. So to edit in 2D, we just click on the. So before we were editing in 3D on this point, for example, here. So to edit in 2D, you can click on this. It has a ring and a pencil button. Just click on that, and it will bring up the 2D editor. So we can then add. If you read that paper, the GSK paper, adding cyclohexyl group at this position actually improves the potency of this molecule. So we added it here. You can see on the right hand side we have this property monitor. So molecular weight, high number of hydrogen bond donors. Green represents good properties for drug likeness. And so once you've made your modification, you just click on the you commit using this button here to 3D. As this button here, and then you can see is that cyclohexyl has been added here and it's heading towards that shelf that we're going to use for designing potency and specificity in the bromodomains. And you can see the score has improved quite dramatically to -29. We can add this to our table of modifiers to save for later to look at. Okay, so there are other options in this Ligand editor in the advanced tab, for example, which we'll cover in some of these we'll cover in 2 weeks' time. And then the last webinar for bioisosteer scan, for example, you can scan different heteroatoms around your ligand. You can find exit attachment points if you're looking to build PROTACs, for example. And there are other options here. So if you're happy with your modifications, you can then save this project, go File, Save Project As, and then you can send it to your chemists or your colleagues, and they can see exactly what you have here. They won't be able to do the editing. If they open it in ICM browser or iMolView, then they'll be able to view exactly what you see here. And so you can annotate, and it's fully interactive, so they can double click on the table and it will load the new ligand. So that's the same. You can also export in file. If you do save as, it will save it in the ICM binary format, which you can look at iMolView or in ICM browser. But if you want to export it as JavaScript, you go to Export as ICM JavaScript. There's a webinar on the JavaScript as well. So if you're happy with your final modifications and you just want to export it as a PDB file or something like that, you can just click this X button here. And then it reassembles the complex and you can save as PDB and just clean everything up if you want to do that as well. Okay, so I just want to thank you very much for your